The aim of the following procedure is to demonstrate how to stain the lysosomes of two types of samples, mouse embryos and differentiated stem cells, with LysoTracker dye to detect patterns of cell death. To prepare embryo samples, embryos are first surgically isolated and placed into a dish. To prepare differentiated cells, embryonic stem cells are used to create embryoid bodies. These embryoid bodies are cultured in suspension and then grown as a monolayer on a chamber slide. The embryos or cell monolayer can be incubated in LysoTracker Red and are then rinsed and fixed. Samples can then be photographed to examine and quantify the staining. Results are obtained that show LysoTracker staining is a simple procedure that can be used to assay programmed cell death in embryos and cultured cells.
The main advantage of this technique over existing methods, like tunnel analysis, is that because LysoTracker dye is a small molecule, thick tissue can be assayed quickly and easily.
Though this method can provide insight into patterns of programmed cell death in mouse embryos and differentiated cultures, it can also be applied to other tissue types, such as chicken embryos and differentiating adult stem cells.
Pair young female mice with a stud male and on following mornings check for a vaginal plug, which indicates that mating has occurred. Noon on the day that the vaginal plug appears is effectively